Okay, so this what we are going to do today. According to this schedule, we'll revisit this page and to see how this projection function is working, and also using a rotation matrix to rotate some points in the space. So for you to understand this, then we will look at using covariance matrix to compute the principal components of PCs. Then we will look at the built-in function of PCA in MATLAB. So how can this built-in function reconcile with what we understand about the procedure using covariance matrix to compute the PC? Then we will look at the t-SNE function. We mentioned how the t-SNE method is very different from PCA, but it's applicable, more reasonable to apply to this complex data structure to look at the relationship between data points. We will also run UMAP. UMAP you will commonly see in the papers. People use UMAP to visualize the cells. Then look at the function of using t-SNE UMAP in the toolbox. We will run with some examples today, and then we can try PCA on the expression matrix of single cell and see what we can get. We also has fate as well, so let me add this in. t-SNE UMAP and fate. Remember, we have this. We will try all this. Using the toolbox to do dimension reduction. Okay. I also have two links to the YouTube channel, YouTube video, and this is an introduction of the meaning of this covariance matrix and the PCA. Will help you using someone else's explanation to help you to understand the PCA and the calculation. This is also a very good lecture to introduce SVD. So SVD essentially is a PCA. A PCA can do through SVD. So if you want to know this decomposition method, and you can watch the video. He's going to talk about this rotation matrix and also how to do this decomposition. We will come back to the decomposition several lectures later, but if you can watch this, we'll give you some good idea. Okay, so let's do a MATLAB exercise to revisit basically this page. I hope you already have some good idea about this projection. So this is most important, because if you know how to compute the projection, then you use this projected vector B as your new coordinate system x-axis. Then you get the position of the A on it. This will be your new coordinates. So I'm going to give a small example to using MATLAB to show this relationship. So let's see, A is a vector, okay? Just do it's like 0.3 and 1.2. So I transpose it, so it's vertical. That's our by default the definition of a vector. So A is in this position. Let's see, B is another one. 0.2, 0.3, also transpose. So I can draw these 2 points on the board here. So if you have your x-axis here and y is here, so A is 1.2 and A is 0.3, 1.2, so A is here, okay? So that's your A. That's your A, and then your B, 0.2, 0.3 is very short, 0.2, 0.3. So B is a very small vector here. That's your B, okay? So when you see A is here, you're using this coordinate system. This is 0.3, this is 0.2. So right now, the coordinate system for current, so this is 1, 0, 0, 1. So this is your x-axis. This is your y-axis. This is your current state, okay? So if you use this as your rotation matrix, see this as your rotation matrix. If you multiplicate this with your A, you will get this number again. There's no rotation. The A is kept still the position with these two numbers. So that's what A is here. So if A multiplicate with 1, 0, so you have to put this into columns. 0, 1. Let's see if this is correct. So this is my current. So I need to put the 0, 0, 1. Yeah, so this is my current so this is my rotation matrix, R is the current. So this matrix will not change, this rotation matrix will not change A at all. So if you multiplicate A with this rotation matrix, R times A, okay, 2 by 2 times A. Okay, so I hope you know this is, I apply a rotation matrix, try to rotate A, but because my rotation matrix, based on this definition, there's no rotation. It's just get your original results here. But now I'm going to use a rotation matrix which defined with a B included. So this B is that direction. Let's use a B as your new axis as X. And we need to find another direction which orthogonal to it as your new Y, okay? So the B I defined as 0.2 or 0.3, is that right? So my B, I just randomly select one. Can I use this as a direction? The reason it's not ideal because this is too short. It's not on the unit circle, so I need to normalize it. So how I know it's not on the unit circle? Because the magnitude, the length of this is not 1. I want to rescale it, make it to 1. So if I look at the norm of this B, it's only 0.3. So I need to find a way to make it longer in order to use this as a unit vector. So I will make this longer. How can I make it longer? I just divide this B with this norm. Now, I normalize my B vector to make it longer, make it into 0.5547, 0.8321. So now you can calculate the norm of this. It should be 1 because I already normalized it by its length to make it longer. So this is normalized B. Now the norm B is 1, okay? So when I write this part is a B divided by its norm. So it's exactly this part. I hope I can draw on the screen. So what we're doing is this part, the B divided by its norm, okay? So we have B divided by its norm. Now we can use this to multiplicate with A. So once we have A times this normalized B, it becomes A norm times cosine, which is the length of this part, this projection. That's the value we want. We want to have this projected onto this vector, and let's see how long this is. Okay. So the length of this is just A times B, as long as B is normalized. So now our B is normalized. So if I A times B, let's see, B is this, A is, so I need to transpose in order to do dot product. So I can either transpose A times B, so A is 1, 1, 2, and B is 2, 1. So this should all work. So this is how long this A projected on B is. The AX coordinate is 0.3 because depends on your original coordinate system. This is very short, it's 0.3. But once you project this onto B, it becomes longer, it becomes 1.16. This is from a dot product I compute from A times B. Okay. If you still have not get there, please ask. Please let me explain that again. Okay. So dot product. So if you don't know the dimension and you don't know how to transpose, then you can just do dot. And the algorithm, the function will automatically put the dimension match and to get the inner product of dot product AB. Yeah.
Could you scroll up to where you normalized?
Oh, normalize the B. Yeah, that's right. That's important because you do need to do the normalization to make this work. Otherwise, you have to normalize that again later. Yeah. Okay. So when I multiplicate with the B, I get the new projection on the x-axis. So this can be used as a coordinate for x, for A after rotation, basically. But you don't have the number for its y coordinate. So now this A becomes 1.1649, and then we need this to be transformed, and its y should be projected onto this coordinate, this vector. So we will do the projection of your original A, the point, to here. Okay, you check how long this is. We got this length, but we need that, right? So this part is also a product, the dot product. We need to have, it's not good. So you project this to this vector. So as we said, this vector is B orthogonal to B. So we need to find this vector here. After that, we can do A dot product of B primer, as long as the B primer is normalized. So for these values here, because we know this is a B for this direction, its orthogonal is to go the x, y, x become negative. So orthogonal one is 0.8321 negative and 0.5547. So this was the original B before normalization and the normalized, and this is orthogonal. So it's orthogonal. Let's see. Is this. So this 2B, B itself and this orthogonal make a pair. So this pair, we can put the B into a rotation matrix. So the B will be here, the left will be B. How to write this? Now the side will be negative B2, B1. So I just manually create this because I know these two are orthogonal to each other. So that's my new direction of x and then my new direction of y. For a given data set, a given data A, I can do the rotation using R times A. Let's see. What is my A? A is here. Yeah, so I use rotation dot product to rotate A, and that's my new coordinate for A on this position. So if you use this as your new XY axis coordinate system, and you translate the coordinates for A into 1.4649 and 0.41. Six. Okay, the same position, A becomes A primer. Depends on how you view this. You rotate your XY, and then you get a new coordinates. And this 0.3 become longer because you project on it becomes longer, and this one becomes shorter because you projected on relatively short. It was here very long after the rotation. So that's the process. You just do the dot product using rotation matrix. Here define the new direction of your XY coordinates, and then multiplicate dot product with A. So I transpose A in order it can be multiplicated. Otherwise, I get this error message. It's not matched. But after rotation, I may want to transpose it back if I want to keep the vertical notation as a point. So let's see, A is this, your rotation matrix is this, and then you rotate it. I want to see if I can do yeah, so that's a way to rotate. Now, so this is only one number. If I have a multiple point, so A is only right now is 0.3, 1.2. So this is my first data, okay? Then I can add another point, whatever number, 0.4, 5.6, or add another number, just some random number, points. $1.2. So in this example, I have 3 points. So this considered to be A1. A1 is here, then I have A2 will be out of the chart here, so 0.4 somewhere, so another A2 somewhere there, I have 8.9, 1.2, A3. So I can have many points on this space. The point is I can rotate all the points just with one operation. So I can do transpose A, so this becomes 3 by 2. I can rotate. So in this case, the same equation. So remember, I use A times this rotation matrix, R. R is a 2 by 2 matrix. But now I can rotate all 3 points at the same time. So the first row is still, after rotation, that's the coordinate of your first point. It was here, it was this, but now the new number is that, according to this system. And the second row is the second point. It was here. After rotation, you get 4.8, 2.7. For this point here, after rotation, you get 5.9 negative. Then look at this. It's become that position. Yeah. Yeah, so it's R is I defined here. R is just 4 numbers, and these 4 numbers indicate the direction of your x-axis and direction of the new y-axis. To make this become a rotation matrix, you just make sure the norm, the length of this vector is 1. Length of this also 1, and these 2 vectors are 90 degree and also orthogonal to each other. You can, as a no rotation will be this. And then once you define a rotation matrix, this applied to all the points once you do the dot product. Okay, let's play with this. Give it a try and see. Can you type this in and try, and I will keep explain each step what that means. So this is a data matrix, contains 3 points. Okay. So first row, 2.2. For 3, 3, 6. Second row, 8, 6, 2, 2. So 3 random points in a 2D space. Then we are going to centralize. Centralize means those data points may be skewed away. So most of these numbers, for example, I define those, they're all positive numbers. But I want to put the origin in the middle of them, so they balance, some positive, some negative. So the average is 0 from x direction and also y direction. So what we can do is just do the average of the two variables. So the variables is on the rows. That's x and y. So that's what we are doing. We do to get a v, which is a normalized version of data. Mean and your data minus 2. Okay, so this is a normalized data. Nothing changed. Everything is in the same position. The only difference is we move the origin 0, 0 in the middle of these 3 points. This is important because we want to compute the covariance matrix for this. And V multiplied by V transpose. You know how to do the matrix multiplication. So the V is 2 by 3, then you transpose become 3 by 2. And the product of this become 2 by 2. So this 2 by 2, that's the relationship between two variables. Two rows, the covariance matrix. And you need to divide this number by 3, okay? By 3. You can put a dot in there, indicate their element based. So every element in this 2 by 2 matrix divided by 3. You don't have to, you just divide by 3 because it's a scalar number. And the program will just divide every element to 3. So this is covariance matrix. As I showed you in this corner here, if you use the built-in function covariance matrix function to do this, you get the same value. And you need to transpose V because covariance matrix take columns as a variable. So you want to keep number of variable to be 2 and sample is 3. So that's what you get. Okay, for these 3 numbers here, we define direction. So this the vector which indicated by this bar here. So we are going to use this new vector E, small e. Use E, so let me define this E, 0.2813, 0.9596. Remember, this is a vector, so the E is this. So I'm going to use E as my new coordinate system, the x-axis. So I need to project all these points onto E in order to compute. So after rotation, I use this as my x-axis. What's the coordinate on x-axis for this number, for those three vectors? So that computation is this, okay? The projection. Project all the points onto, just like what we did here, the project this 3 points onto B. But in there, we project 3 vector, V1, V2, V3 onto E. Okay? So I want to get the projection V transposed times E. So these are their projection, how long their shadow basically on the new vector. So this length is what we need. We need to compute the length and also square them and the summation. So this gave you the variation, basically how much the difference, how far away them away from origin, basically. So we computed this number because we want to find a E. So if you define the E differently, you will get different values of this. For example, if I generate E, let's see whether E is normalized. E is normalized, so E can be used as a direction. So I can change E to any random number. So right now, this particular E gave me this. If I define another E2 as for instance 0.55 0.1. So I can randomly pick 2 numbers and then normalize it as another vector to show the direction. Okay, so I can use this as my coordinate system as my x-axis. This is different from the E defined, but let's see if I rotate the data to E2, I get, is this bigger than the E? Is this yeah, so E2 is better in this case. You get more dispersed projection values on E2. Okay, so the PCA is trying to find the combinations of these 2 values. So now I tried 0.55 and 0.1 as a direction. For E, I tried 0.2 and 0.95 as direction. Looks like I should find a projection position, for example, should be this direction. Okay, it should have a vector on this direction in order to let this 3 points to project on this vector that gave them longest shadow. So if you do the PCA for these 3 points, you probably will get the first PC in this direction. So that's this the process. So in order to find the so in this example, I also show you can use this to compute the variation. Okay, it's the same as so for E, you get the same value if you do the covariance matrix composition. So I haven't defined the C yet. So you get the same number as your compute the summation of all the projection. So this just illustrate that you can use covariance matrix and multiplicate this easily with your new vector, your selected direction, and to get the total length of projection of all the points in your sample. So because these two numbers are the same, so if you found good E gave you the maximized this value, it is the maximized this value as well. So we need to maximize the expression of this. And this can be maximized by using the eigen decomposition and to find the largest eigenvalue and its coordinate eigenvectors, and using this largest eigenvalue corresponding eigenvector as a E that gave you the largest solution for this. So that's why in the procedure to compute the PCs, we use, in this expression, we use covariance matrix in order to find the rotation matrix, give you the maximized dispersion of the data points. Okay, in this stage, let's try clean this. Let's do the doc for PCA, the function. So PCA is a function provided by MATLAB, and so which, if I search which PCA, you can find is inside a toolbox of statistics. Statistics classical methods, including PCA as a function. Remember, you know how to write a function, and or the function end with a .m extension. So this is a definition of the PCA function, which is there located. So when you call this function, the program will search the definition of that function and give you put x in there. X is your input. Okay, let's look at the syntax of the PCA function. So you need to give a matrix. That's the simplest way, and it will return the coefficient. So your input is a data matrix, which n by p. Remember, n is your number of samples. So p is your variable. If you translate it into vector definition, so your row will be, each row is a vector. So normally we put a vector as a column, so you have a vertical column vectors. But in this case, you need to put your vectors in rows and stack them in order to give PCA. But because that's by definition, this is like statisticians, they are using rows to collect samples, and the columns are the variables to explain the samples. So if you gave the expression matrix for single cell data, remember normally we have genes in the rows, columns are the cells. So, but when you apply PCA, you want to do the dimension reduction for cells. So cells are the samples. So you need to transpose your X matrix to make rows are the cells and the columns are the genes before you give to PCA function. So just make sure you understand your dimensionality of your sample. Don't give it wrong, because you will get rotation matrix in the coefficient. Okay. So if it's 2D data, you'll get a 2x2 rotation matrix. If 3D data, you will get a 3x3 rotation matrix. Inside the algorithm, there are different ways. Several algorithms have been implemented in the PCA built-in function. As we explained it, we use covariance matrix and to do this eigen decomposition. This is one of the methods implemented in the PCA you can require. But if you don't require the PCA function, we will use SVD algorithm to do the same thing. So we can try example in this provided. Okay. Let's copy this part. This will load a data and called ingredients. So this is a built-in data set used as an example data. So let's clean my workspace. So I have nothing in the memory here. Then I load this. Then we have a 13x4 matrix called ingredients. Those just 4 vectors of numbers, but each row is a data point, is one sample. So this is your input. So if you do this, PCA apply to this, and you get the coefficient. This is the rotation matrix of your 4x4 rotation matrix. The reason is 4x4 because you have 4 variables. Okay, each define a property of your sample with respect for some properties. One of the 4. So this is your PC basically. So if you need a rotated data point, you can see, okay, I want to get a score. Okay, so this will return not only the rotation matrix, and also the transformed location. Remember, your input, this ingredients, is 13x4. And now the score is also 13x4. This 13x4 is after all these 13 points have been rotated, and you get a new coordinate for all the original positions. And this is after rotation, what you get. Okay? So in this case, we can start to visualize using a scatter plot those points. The first and the second. This is like 2D. So that's where the data are located. There are 13 points. So this is after rotation. And display the most separable perspective. So if you don't do the rotation, then you can use the gradients, the first and the second if you do this figure, this is before rotation. You get the first two variables, separate samples look like this, and so this is original data scatter plot with first two variables. You can do 1, 3, and give you something like this. So this is before, and then after rotation, you get using the score to do the first two. And you can also plot after rotation score from dimension 1, 3, 2, 3. You have combinations, but you know the first two gave you the masked separation for the points. Okay, so let's take a 5 minutes break, and you can spend some time on this page. Read the documentation and see what you can get from this function. So when we come back, we are going to use this small example, ingredients still. Let's do it using covariance matrix method. Let's see if we can get the same rotation matrix, same kind of projection, okay, without using the built-in function. Yeah, I hope everybody sooner or later can get your head around exactly what happened here. If you still have some trouble, you will get in troubles in explain for the homework, take a video, and if you still have a problem, then you won't be able to make it. Because if you don't understand this coordinate system change, rotation, how the PCA finally get the rotation matrix out of the data, will be really hard. But that's really, really important. That if you forget everything from this class and you get some of those ideas, it will be very useful later for you when you do the data analysis. When you apply PCA to your data anytime later in your career, to understand how this eventually happened, how every steps you can at the end to use rotation, use multiplication of matrices to achieve. So you can decompose every steps until you reach to the multiplication between two matrices. Okay. If you still have trouble in these days, that's your best time to learn this. We have a TA in the VRB building, 366. Anytime you come, and the Shran should be there. Just let him to explain to you. And my office is 382. Let's just using the whiteboard, blackboard to do that once again, and they should be able to get it. Just make an appointment if you want to come, okay? And the 366 it was a white lab, but now converted to the computer lab, so we have a lot of space. If you want to spend half a day in there and chat with my lab members, feel free to do so, okay? So in our case, we want to not only use the PCA to get the rotation matrix or rotated score of the projection, but we want to do manually to see if we don't use the PCA, the built-in function, can we get the scores, can we get the coefficient 4x4 matrix? Follow the procedure we described using covariance matrix to do so. Let's see, if we have the data, let's just give this name as big X again. So ingredients. Okay, I just changed the name so easy to handle. So this is a matrix with 13 samples, 4 variables. We want to follow the procedure described here. Okay, we do remove the mean first. The mean is for every variable. So for first column, what's the mean? For the second column, what's the mean? And we want to every values to remove that mean. So to do the mean, we compute mean X. This will give you 4 numbers. Each is the average of your each variable. So we can see the second variable is on average 48, is larger than first. And then the largest is in the middle. And that's correct, right? If you have an impression of 4 columns. So now we remove those means by X minus mean X. Okay? So this program, this coding, the language is smart enough. It will remove every element in the fourth column by the mean, the first mean here. The second column will remove that. This is a new feature of the language. If you do this 5 years ago, maybe you have to handle it differently, but now the program is okay. So you just get, after you normalize, you centralize your data, you save your centralized data back to variable X. So now X is centralized. Maybe I should put centralized C here, okay, rather than original. Okay, you start to see the negative values, some of them. If you do mean right now, what should you get? What's the column, first column, the mean? So they centralized that everything should be 0. Okay, the mean again, because you moved. And they are not mean, it's some numeric bias, but it is a very, very small number, so it's almost 0. Okay. There's some precision of the machine, so you have it using 60 bits or 32 bits. It's still not absolutely 0, but it's very, very small already. So that's what we centralize the data is like this. So what's next? We centralize the data, then we compute the covariance. So you need to transpose x times x sub. So now it's xc. Let's see, xc is 13 by 4. Now we need to transpose itself, become 4 by 13, and then multiplicate 13 by 4 itself. So this is a covariance matrix. Now you need to divide by 13. So this is the C. This is the covariance matrix. It's 4 by 4. It's symmetric, and also every number is positive. Every number is a real number. There's no complex number here. If we do a covariance matrix for x, then you put 1 here. Let's see if we can get it. So we get the same thing as we compute with a built-in function. Okay. So now we confirm we have a C here. So what's next step? We are going to do the decomposition, eigendecomposition with the C. So to achieve this, we need to get V and D, and this eigen C. So this function will return the eigenvector and also eigenvalues. So we are going to use eigenvectors, which has its corresponding eigenvalues. The largest has a fourth component. What this gives to you, if you look at the 477, this last one, so the last column is your rotation matrix, the first axis. Okay, so your second PC, the second coordinates, so this is your x-axis, this is your y-axis, so this is the last column is your x-axis, this is y-axis, and then third, fourth. So according to this, we need to put 1, 2, 3, 4. We need to rearrange the vectors. So this becomes the first column will be 4, 3, 2, 1. Okay, this is our rotation matrix. So this expression will extract the last column first, and then third column. The third column that do nothing. It just shuffle the rearrange the columns order. Okay, So this is our rotation matrix. Let's compare with the coefficient. So this is the coefficient computed from PCA itself. So what we get here is almost the same as what the built-in function calculated. So this is our rotation matrix. And for the first rotation, first column, which gave you the x-axis, is the same. The second one, slightly different, is the direction, okay? You see, this is negative, and this is positive, the rest of positive and negative. So basically, that you have x-axis is fixed, then your y-axis either go this way or that way. There's no difference there. It's just direction. And the third is the same, and the last is the same. So as you can see, we followed this standard procedure to compute the rotation matrix, which is the same as PCA obtained using the built-in function. Okay. So how we get this is from x, you get the same score as your computed step by step. The score is, so if you use the rotation matrix to rotate your data, rotate your original, I don't know how to do this. So x is 13/4. So let's transpose x times r and then rotate the back. So this will be your rotated r, x arc rotated. So x is, let's do this. So that's apply the rotation, rotate your original data, and you get a rotated version of your original. This should be very similar to the score. Yeah, for some difference, I have to check why the, so did they rotate it to the C, maybe rotate the centralized version. Yeah, so then you rotate the centralized version and you got the score the same as the function created. Okay, we have to move on to Ranticini function in MATLAB, okay? So you know how to get the PCA, and that's documentation PCA, okay? And you know what x should be, what y should be. Now your job is, we have a built-in function called TSNE. Please let me know where the TSNE function is located, and do you know how to get the documentation for TSNE? So what we should type here is which, okay? It's still in the same toolbox, same location in the PCA, it's the same place. And then if you do the doc TSNE, and you'll get this page to explain to you how to do this TSNE analysis. So this will take a matrix with two return, and then take high dimensional X, okay? Will return a matrix of 2D embedding. So basically we can call the same, we have X here, okay? So this, we can just TSNE, put X same as a PCA. Now it return the embedding, which is the 2D positions. That's it, okay? It's very easy to use. And if you look at the Y, the return is just 2D position for 13 samples. You know how to calculate, how to figure scatter, and then you have first and the column and the second column of this distribution of the data. This is how you use TSNE. This small example is not the best to use as TSNE, because TSNE works really good for even higher dimension. So, but the X only has 4 variables, so the 4D data reduced to 2D. So this is not the best data. So I'm going to, so you know how to use TSNE to reduce the dimension of a given data set. X. Now I'm going to use a real UMI, gene expression matrix, from single cell data. And you can do this and see whether you can generate a TSNE plot from the single cell data using this command line, okay? Let's do this. Let's just load example. So let's see where you are. You should be in, let's see, example data. So this is a little bit long, but if you can use the tab, you should be able to load this correctly. Now after that, you can do sce dot x. Remember to put a semicolon at the end. So if you can find a way to type this to make this work, and you should get an expression matrix, a real one, from this example data. Now I ask you to do the TSNE embedding for this expression matrix X. That's your job, okay? Then get the expression matrix assigned to X. So this X is a huge matrix with thousands cells and genes. Now you are ready to do the TSNE and then make sure. Let's see what you get, okay? You didn't put this in there? Oh, use Control C. You can stop. Okay, yeah. Questions?
I guess do I just have to clear everything before then? That's why.
SCE. Oh, you may not have that.
File yet?
You do this. Let's right-click this min. Yeah, right-click. No, this the folder.
Oh, okay.
Yeah. Then you do the add to the parse, and then select the folder. Yeah. Now you do this load again. It's working, and then do the X. X. Yeah. Semicolon. Now you can do the T-SNE.
Okay.
Yeah. So because you have a big X here, it's huge, right?
Okay.
Yeah. Questions? Operation. Yeah, you can Z. Yeah, that's okay. Okay. Good. Plot. Okay. Yeah. So let's see if you can have a scatter plot. Any questions? Not a name. You don't have SC in there, so you haven't loaded that in. Oh, you do this. Let's go here. Let's do that load again, because you didn't found the go up. Yeah. No, go down. Go. That's right. You don't need the last. No, you don't need this. Yeah. Let's just do a tab. Click return. Okay, then run this. Yeah. Then do the T-SNE and the scatter plot. Yeah, you may get the scatter plot, but make sure. Remember, this is the X itself is a gene by cell, but the function of T-SNE require cell by gene. So you need to do transformation before you can do anything. Okay. Example data. What I put there? New. So I just do the tab. It will automatically load that in. Then you can do the X. Yeah. Yeah. Get the figure, but not yet. So to make this right, you need to transpose X. Otherwise, you get a scatter plot for genes. Before you send it to TSNE function. Yeah, so you can have, I think now, yeah, it's better to give to another variable, because otherwise you'll change the X. Yeah, you may want to change it to the name of Y. It's better. But now your X has been changed, so you have to redo this to get X spike. No, you just do this, go up. And then, yeah, so now your X is back. X is this big one. Now you do TSNE, but not assign to X, because you want to keep X. Now you can put a Y, something like a Y, or but you have to transpose X first. Put it this one. This is correct. Yeah. I think you are getting the right one, and you get the right one. Zara, you get the wrong one because you didn't transpose. You get this is a gene. Each point is a gene, so the correct one should be. Let's see. Do the transposition. So just transpose the flip the cells genes. Yeah. It's different because you this is stochastic. There are random numbers in the between. So if your machine is different from others, then you need to So let's do transpose this.
Okay.
Yeah, do that again. Unless you really want to study the gene-gene relationship. It also makes sense sometimes. We do this. But for our purposes, if you get something look like that, the cluster of cells, that's right. Okay? So make sure also so I reset this random number seed in order to can regenerate exactly the same location, reset this, because the stochastic process in the middle of the TSAE, the random generator to pop up random numbers, follow some of those distributions that we explained it. So we cannot guarantee each time you get the same figure, but if you say, oh, I reset my random number generated seed to the same states, then the random number you generate in between will be the same. So we can get exactly the same plot. You don't have to, the biology should be the same in all this. Okay, so that's what we get. You can count how many cells in your X matrix. Remember the cells in the column. So the number of those, because you transformed already, each point is a cell. Okay, if you, there's no built-in function for UMAP, but I do have run MATLAB UMAP. So ML stands for MATLAB. So this is a built-in the function I developed, not I developed, I just incorporated from other implementation. So this will take not transposed, okay? So this will take y. You put a dot run, so this just use this as a built-in function. If you have the toolbox installed, this should work. But this time you don't have to transpose X, because this function I designed to allow it to take genes in the rows, cells in the column. And then you get Y. So this is UMAP. You don't have UMAP. Oh, there's a gene, yeah.
I don't know why mine so comes up as this.
It's very different from what we have, all right? Yeah.
I'm not sure. I tried the other way too.
Uh-huh.
And when I did without the I had forgotten to transpose.
Uh-huh.
And when I did it like that, it just.
You still have this.
Yeah, like this is what I had.
Uh-huh, yeah.
It could be something else. Right?
Yes, let's try, let's see, X is 7,000 cells. I think you transpose, I don't know, you transpose that. Yeah, so because you transpose that already, and you saved. So now you can just do this directly. If you look at my dimension of my X, I have 7,000 rows. Okay. But now you already transpose.
I see.
Yeah, so you just need, yeah, do this without, and then get a figure. But for the UMAP, you require, so you just make sure you know numbers of cells, genes. Yeah. So I don't know if you can get the UMAP working.
Oh, it works.
It works. Okay. So this is your t-SNE, and UMAP is similar. You get a different. So this is my UMAP embedding. First two dimension. Okay, you have both. That's right. That's right. Someone, my collaborator, like UMAP because they're more clear and more how to say condensed presentation. But I always found the shape is weird, so I don't like. That's right. Yes. Yeah, UMAP. Yeah, in this case, when you do the UMAP, you have to transpose that back. Yeah.
I like this is.
Yeah. Then you get. Yeah. You don't have to copy. I always use the up down to get. Yeah. Now I encourage you to use the PCA to do this. Remember you know how to use PCA to do the dimensionality reduction. So that's the PCA, right? And this is Y. There's a projection we need. So the score is Y. We don't need this rotation matrix, just like the function to rotate it and give me the results, the projection. And make sure in this case, it's either by cell or by gene. So now X is by genes in the row, and the column is the cell. But the PCA function requires your transposition. So we need to do this, okay? Let's see what you'll get in this. That's my PCA result. The first two dimension. Yes, you've got this T shape. Yeah, so that's the reason people start to use t-SNE or UMAP rather than the PCA itself to do this. Yeah, you get something different. This may be for the genes, maybe, I don't know. Yeah. So if you do this correctly, let's see, you should get 2,000 cells, something like that, points. So your Y should be 2,000 something because we have 2,000 cells. let me see. You haven't stopped yet. Still busy.
So,
Yeah, it's done. Yeah.
Okay, so just go to the figure then.
Yeah. Same one. Do you need a link to the video recorded? You got the link? Okay. Even for the Tuesdays? No. I don't know if you have a whole channel. I will find because Tuesday you were not here.
Right, yeah.
Yeah. So I will send you a link. Okay. Mm-hmm. Okay, any questions for which one we missed? We still have one more. Disney UMAP fit. Okay, let's do the fit. So this is UMAP and feed. Okay, easy. Oh. I will show you the code. Okay, so this is your use same as UMAP, the same input cells in the column. So you don't have to transpose your X. And then when I get this is fade projection. Okay. I think we are still playing with the genes. I don't know. Yeah. Good. It's the same thing. All right. Yeah, got the same thing. Okay. So now I hope you know once you have this expression matrix, you can do using different ways to do the dimensionality reduction. And basically that's it. And for this part, let's look at in the toolbox, how can you achieve all this? So I'm going to do an update. Remember, if you can use the up and the down key to revisit the previous comment line in the history, so you don't have to re-tap. Anytime you can type first the two characters and use tab to find those comment lines in the history to make things easier for you. Here I'm in the right folder, so I'm just updating my folder of a toolbox because there's some changes over the weekend. You don't have to do this. It's a very minor change to update your toolbox to the latest version. Yeah, it takes some time to upgrade. Do you have any questions in the meantime? Okay. Okay, so in the last 15 minutes, we will use the toolbox to achieve all this kind of embedding from the very beginning. So in this case, I'm going to use example data to load example data and use the raw data. So this is just like you start with from scratch. So you download a data without any annotation yet. So now there's no embedding. So all the cells have been put in this 3D randomly. They're distributed, normal distribution. And in the center, this position 0, 0, 0 have the most cells in there. Remember the shape of the Gaussian curve. Okay, in the middle, you have a lot. So this is 3D. Now we are going to use this button here to do the embedding. And if you click this, you have options. I will select them all. And then including T-SNE map and the fate, 2D and 3D combinations. And just like this to do it. This is like highly variable genes, 2000. So the interface will go through all this different methods and combinations of the parameters and generate 2D, 3D, everything. So you have all the embeddings for the same data set. So 2D is easy to publish, to copy the figure and put in your paper. 3D help you to have one more dimension to visualize the cells. Maybe they're not very separable in some detailed position, but if you use 3D, you have one more dimension to separate the subtype of cells. And the three algorithms each have their own feature. So now you finish all the embeddings. Now if you want to see all of them in the same picture, you can see yes. If you don't see yes here, you can still use this big eye icon here later to visualize them. Just see yes. In this case, I have all the embeddings in front of me. If you start from the upper right corner, this is a 2D Disney. And this is a 2D Fate and the 2D UMAP. And you have 3Ds. Using this, you can start to click this brush here. In any of them, if you highlight some of those cells, in one of the figure, you can see their corresponding position in other dimension reduction technique. So for example, this group of cells looks like an independent cluster, but in Fate, you can see it's attached with something else. I don't know which group of this is. I can start to clear, select this. Looks like this group here. These two group, which I highlighted in this. So these two groups, for example, here, these two groups, they're isolated from the Disney embedding. But in Fate, it creates a triangle here, okay? This triangle, a lot of cells, looks like it was embedded in different clusters. And also these two groups looks like they're linked somehow in the Fate. And in Fate also you have this long, so these two are linked. And UMAP and the Disney are very similar approach, so you get highly consistent clustering. But the problem for the Disney UMAP is they treat these small islands. They are independent, because they are generative model rely on the high dimensional distance, whether they are close to each other or not. Then when they project the relationship into 2D, if in the high dimensional space, these two cells, two group cells, slightly away from each other, it may reflect in the 2D far away from each other a lot. Well, Fate is very different algorithm. The cape the distance matrix information. So in some case, for example, these two, if you only rely on UMAP, you may think about these two group of cells are highly isolated. But in reality, fate suggests they form a branch here, and also they may linked, they may cell state change from one state to another. So if you don't have that information, you will not consider these two groups are related. So that's why I encourage you, once you have your own data, look at if using different technique and visualize to thoroughly understanding the relationship between your subtypes of the cells. So this is one way you can do it. Okay, from here, for example, this is a 3D fate. If you want to change, you can say, okay, I want to use existing, and then select this, because you already computed it before, so you just replot the relationship. Okay, so that's what we achieved this week. To revisit that page, you click this big I here, okay? Then multi-embedding. This will allow you to select, you can select them all, or you can select several methods in order to do this one figure of all embeddings. And then you can start to brush to do those detections. You may want to show the expression of some genes, then in this case you can find genes, and this particular gene's expression will be showing on this multi-dimensional multi-different views. And then, for example, you can look at the bar. The red indicates the high expression, and the gray indicates there's no expression for this gene, ABCD3. So this gives you some very quick view about the feature genes. If you know some marker genes you really want to study, and you can have a quick that's INS. You can have a quick understanding. For example, this cluster have a high expression of INS. This is insulin genes. So this most likely to be beta cell. If you look at the bar here, expression level really, really high. You can also look at the library size. That's how deep you sequenced of each cell. So these cells have been sequenced more than those cells color-coded. You can also look at number of detected genes and others. Okay. There are different phases of the cell cycle. Okay, that's all for this week. Let me know if you have any quick questions before we finish this session. Oh, I will see you next week, next Tuesday. Keep warm, everybody.